Hello, everyone. My name is Doran Parker, and today I will be talking to you about LSD and its effects on mice behavior. The first thing I want to talk to you about is going to be the history of LSD. It's important to know the background, the history, and how it was made. The next thing I'm going to talk to you about is chemical structure slash effects. I'm sure many of you know what the effects are, but the chemical structure is also very important. Next, we're going to talk about mechanism of action, how it works in the human body. Then, explanation of mice as a model system. This is basically explaining why we use mice instead of humans for LSD trials. The fifth thing I'm going to talk about is the introduction of my research paper. Next, I'm going to talk about the interpretation of data from the research paper. And then finally, I'm going to conclude on what the conclusions were from the research paper. And then lastly, I'm going to tell you the take home message that I want everyone to know by the end of this talk. So let's move on to the history of LSD. LSD was first synthesized in 1938 by a man named Albert Hofmann. He was a chemist who was working for Sandoz Pharmaceutical in Basel, Switzerland, and its hallucinogenic effects were first unknown until 1943 when Hoffman accidentally consumed some LSD through his fingertips. After that, he noticed the hallucinogenic effects that it had. And it was later found out that an oral dose of at least 25 micrograms, which is very small, were capable of producing vivid hallucinations. And scientists, after figuring this out, were really extremely impressed by the drug and wanted to study it more. So soon after, the study of LSD was skyrocketing in the 1940s, 50s, and 60s, and it increased dramatically over that time as well. Many studies of LSD before 1970 were primarily focused on how this drug could be used to treat anxiety, depression, as well as addiction. Finally, the drug managed to escape the lab and it made its way into the hands of the common people. And this took place primarily in the 1960s. And this spurred a counterculture movement that of people who used and abused it to escape the problems of society that they had during the time. America was just in war with Vietnam. People were heavily against the war, and people just really didn't like the government, and they wanted to use LSD to escape all of these fears and anxieties that they had about their society and their government at the time. And so it became very popular in pop culture, and it was heavily referred to by the music industry at the time. For instance, the song Lucy in the Sky with Diamonds by the famous, well known British band The Beatles is a reference to LSD, Lucy being L, Sky being S, and then D being diamonds. Shortly after, the Western intelligence community and the military saw it as a potential chemical weapon, and they even said that it was capable of mind control. I don't know whether the validity of that is true, but they decided that it needed to be banned in 1967, and the drug was perceived as a threat to public safety. This is why it was banned, and global stability as well. And furthermore, in 1970, the Controlled Substance Act was also passed, which ceased all the research being done with LSD and hallucinogens. So it wasn't only banned on the streets as a recreational drug, it was also banned within research. And with that, let's move on. So here's a quote by Dr. Stanley Yoles, who is the director of the National Institute of Mental Health from 1964 through 1970. As I mentioned before, this was a time when they were studying LSD heavily. And I wanted to bring this quote to show the impression of some doctors who were working in the field of LSD during that time. He said that it's an important drug in treatment of mental and emotional disorders. And clearly, we had scientists who believed that there would be positive correlations to LSD and its applications in medicine. Now we're going to talk about the chemical structure of lysergic acid dithylamide, LSD. The molecular formula of LSD is C20H25N3O. This means that LSD has 20 carbons, 25 hydrogens, 3 nitrogens, and 1 oxygen. The L in lysergic or lysergide belongs to a family of indole alkylamines. that includes numerous substituted tryptamines, such as psilocin, which is found in magic mushrooms, as well as NL dimethyltryptamine, or by its common street name, DMT. And these are two other hallucinogenic drugs that are fairly popular amongst the hallucinogenic drug category, along with LSD. And for those of you who are wondering, LSD's IUPAC name is 910 didehydro NN diethyl 6 methyl methylgoline 8 carbomaximide. So try to say that 5 times fast. So essentially, the lysergic acid dithalamide is an ergoline alkaloid, which means that its structural skeleton is contained in a variety of alkaloids referred to as ergoline. And this is the result of a formal condensation of lysergic acid with dithalamide. A condensation is basically a reaction where two molecules react with the resulting loss of a water molecule. So, just to give you a little bit more information on LSD, repeated administration of LSD over several days promotes rapid tolerance, usually, where the drug has a much less effect than it normally did. The hallucinations people feel are significantly less than from the first time that they did it. This tolerance may be associated with a downregulation or decrease in number of 5-HT2A serotonin receptors, which we'll talk about that later in the slides. And LSD is not considered to be addictive at all. And although it is a potent drug, it is generally considered to be non-toxic and safe in terms of its effects on the body. However, government organizations say that it is not safe, so it really depends on who you ask based off of how good it is or not for the body. Listed as a Schedule 1 drug, and someone could land a 3-year sentence in prison for LSD possession. So, probably not a good thing to keep around in your pockets. For those of you who do not know what a Schedule 1 drug is, it's a substance or chemical with no currently accepted medical use and a high potential for abuse. Furthermore, more uses for it are recreational. It's more so after the 1960s through the 80s, perhaps, where LSD use was very high back then. And it was used basically because of its hallucinogenic properties. And before the 1970s, it was used for treatments, potential treatments as well, for, as I mentioned before, anxiety, depression, addiction, and scientists are still using it in order to see what other things it could treat. And with that, let's move on. Now we're going to talk about the mechanism of action. Although we have some understanding of LSD's activity in the brain, it is not fully understood as to how this activity leads to the subjective experiences that people have and feel when taking the drug. Regardless, it is thought that the LSD's activity at a subtype of serotonin receptor called the 5-HT2A receptor is critical to the drug's psychedelic effects on the body. LSD acts as a partial agonist at the 5-HT2A receptor, and this means that it binds to the receptor and generates a response that is a fraction of what the natural ligand serotonin generates. So it's kind of an excess amount of serotonin. And LSD also interacts with other receptors, although the role of these receptors and the effects of LSD is less clear. For example, LSD binds to other serotonin receptor subtypes, as well as subtypes of dopamine and adrenergic receptors. And we still don't have that much knowledge as to the mechanism of how LSD works, but this is probably the most we have, the most understanding that we have on how it works exactly. The downregulation of 5-HT2A, as I mentioned before, leads to tolerance from LSD to LSD by the 5-HT2A receptor because it's just a constant overstimulation. Eventually, it's going to get null, like it's going to get dull to the effects of LSD back onto it. So that is another way that science understands how the we get tolerant to LSD. So now I'm going to talk about mice as a model system for the testing of LSD. So, although human test subjects have been banned from tests involving LSD since 1970, rats and mice, however, were not banned, and it is through these animals that we are able to elucidate more information as to the effect of the drug on certain cognitive functions and behavior. Although this allows researchers to test a drug, it does, however, pose some drawbacks. For example, rodents have different sensory systems than humans. They have relatively poor vision and comparatively well developed olfactory and somatosensory abilities. Olfactory relates to the sense of smell, meaning that rodents have a better sense of smell than humans. And somatosensory relates to sensations such as pressure, pain, or warmth, which occurs anywhere in the body. This is why one of the main priorities for most research involving rodents and LSD is to determine mechanisms of action for these behavioral outcomes and relate them to humans. So, just a few logistics. Fair treatment is often argued when testing mice versus humans, and a lot of corporations are against it, while a lot of corporations are for it. My study, in particular, does have a mice model for the test, and this is very important for behavioral studies because testing these mice, we could understand from their behavior how humans react as well. Not exactly so, but it's still very effective, and we learn a lot from these studies. So now I'm going to talk about the paper that I have chosen to present. The study was LSD administered as a single dose reduces alcohol consumption in C57BL-6J mice by Kenneth Alper et al. For those of you who do not know, C57BL-6J mice are an inbred mouse strain that originally developed for the study of anti-tumor activity and immunology, and they have been commonly used for other tests as well, such as mine. Just a little background about the author. Dr. Alper is a clinical associate professor in the Department of Psychiatry at the NYU Grossman School of Medicine, and he received his doctorate from the University of Texas in 1984. Throughout his life, he has written many papers and research about various hallucinogenic drugs such as LSD, as well as other synthetic drugs. So he has a very well-rounded background in the study of hallucinogenic drugs, and I see him as a scientist who I could trust his opinion on. Now, on to the research question and the paper hypothesis. The paper suggests that there is substantive clinical literature on classical hallucinogens, most commonly LSD, for the treatment of alcohol use disorder. However, there has been no published research on the effect of LSD on alcohol consumption in animals. So we want to know if this is actually true, if indeed we have hallucinogenic drugs can treat alcohol use disorder. The researchers wanted to test that hypothesis that, that LSD could be a potential treatment for alcohol use disorder. For those of you who do not know what alcohol use disorder is, AUD by its abbreviation, uh, it's basically a medical condition characterized by an impaired ability to stop or control alcohol use despite adverse consequences. And it can take many forms. It could be mild, moderate, or even severe based on the number of symptoms one experiences. And it's often prevalent amongst alcoholics and people who are more inclined to addictive tendencies. So, if we could find a way to solve this issue within people, through the use of hallucinogenic drugs such as LSD, it would be very beneficial for everyone as a whole. And with that, let's move ahead. The effect of LSD on ethanol consumption was assessed using a two bottle choice drinking paradigm. And this is where animals are presented with access to two bottles, where one contains ethanol solution and the other contains a non ethanol beverage. And the animal is basically forced to decide which one will it drink more if it's more inclined to drink alcohol or ethanol in this circumstance. then, or it's addicted to it, then it will continue to drink from that ethanol solution more so than the water solution. So this is basically what a two bottle paradigm means. And the mice were housed individually in cages and were made to habituate to a reverse light dark cycle for at least a week. So they just switched day and night for them. They were then introduced to ethanol to develop a preference between two bottles, one containing water and one containing 20% ethanol, as mentioned before, which was offered 24 hours, 5 days a week for 4 weeks straight. The researchers then divided the mice into three separate categories, equal to the amount of ethanol intake that the mice had. So if particular mice had more ethanol intake, then they would separate them, make them into their own separate group versus ones that didn't have as much. And the experimental group here were the mice injected with 25 or 50 micrograms of LSD, and the control were injected with a saline solution just to make sure that everything was in proper order. And the mice were then put back into their shelters in their cages, and water bottles were replaced with bottles containing water and ethanol after 10 minutes of the treatment, after 10 minutes of being injected with LSD or saline solution. So the amounts of ethanol and water consumption were measured every 24 hours or so, and the researchers changed the positions of the water and ethanol bottles to avoid place preference. So if the mice preferred a certain location for the bottle, whether on the right or left, this would basically minimize any of that place preference so that we can get effective results. And furthermore, two bottles of ethanol and water were placed in an empty cage with no mouse as a control for spillage and evaporation, which is also a very important feature of the experiment because spillage and evaporation could have an effect on the results, and that's why they had to do that. So our dependent variables here, as stated in the slides, are the alcohol consumption, the ethanol preference, and the total fluid, which you will see in the results section of this presentation. Now let's move on. So this is the first data table that we have. This is figure A and figure B. On the y-axis, you can clearly see it says ethanol consumption, and on the x-axis, it's the days of exposure. And this for all the graphs, the left graph represents mice with a single dosage of 25 micrograms of LSD, while on the left represents the mice with single dosages of 50 micrograms of LSD. And remember that the mice were divided into three separate groups of equal ethanol consumption based on the amount of ethanol consumed on the day before the LSD administration. So as you can see in the group treated with 50 micrograms of LSD in graph B, right over here, there appears to be a more significant separation in terms of the ethanol consumption. The ethanol consumption on the left with as the y-axis. You can see a clear separation between the mice in the black with LSD 50 micrograms than the saline solution with in the white. As opposed to on the left, you could see the ethanol consumption for the graph A after the injection of the 25 LSD micrograms. It kind of overlapped just slightly, and it wasn't as clear as separation between the 50 micrograms versus the 25 micrograms because you could see the overlap on the graph in these particular areas. So we see a greater separation with the 50 micrograms versus the 25 micrograms from the saline solutions. Now moving on to graph C and graph D, we can see that this is representing on the y-axis ethanol preference. It's measuring ethanol preference versus the days of exposure. And basically, again, we see a certain trend where LSD that has not mice injected with 50 micrograms of LSD had a higher separation than the mice injected with the 25 micrograms of LSD because there's more overlap in these part areas right here than over here. There are some areas of overlap in terms of like right there and I guess right over here. But as you can see with the naked eye, it's more separation, especially from days 7 through 13, and even 35 through 41. You can see the separation clearly between the two. Although it's not exactly, it's not such a huge separation. It does show a a slight percentage change from the saline solution in the 50 micrograms versus the 25 micrograms. Finally, in data table 3, we could see that the y-axis here is the total fluid consumption versus the days of exposure. And this is important to see because the total fluid consumption is overlapping on the 50 microgram and the LSD microgram with all the saline solution mice. So what does this mean? That this basically means that the overlap is alluding to the fact that the mice still drank as much fluid as they normally do, and the LSD didn't have that big of an effect on the mice behavior in terms of fluid consumption. However, as we saw previously in the other graphs, it did have a clear effect on the ethanol preference and the ethanol consumption. And as you can see, there's overlap throughout all the days from the 25 microgram and the 50 microgram LSD groups versus the saline groups. And with that, we move on to our conclusions. So the study found reductions in both ethanol consumption and preference in the group of mice that were treated with the LSD dosage of 50 micrograms versus the 25 micrograms. The observers found that ethanol consumption was limited with a reduction of 17.9%, and that was sustained throughout the 46 days of the experiment, which is very important to know. Furthermore, the total fluid intake and locomotor activity did not differ among the control and LSD treated groups. The researchers offered potential reasons for this trend. A lot of them are very complicated. One of the stated reasons that's been suggested was that persistently reduced signaling in downstream pathways linked to the 5-HT2A serotonin receptor, as we mentioned before. Um, uh, Through the effects of LSD might explain sustained effects observed in clinical studies on substance use or mood and anxiety symptoms. Furthermore, diminished signaling in downstream pathways linked to the 5-HT2A receptor um, could increase transmission via metatropic glutamate receptors 2 and 3. And it's due to the reciprocally inhibitory relationship between the two receptors that this increased transmission through the glutamate receptors might be expected to diminish the alcohol intake, and it could be a possible a pharmacotherapy solution to alcohol dependence. So, in summary, the experiment concluded that they found that LSD administered as a single dose reduces ethanol consumption and preference in C57BL6J mice, and that these findings indicate an apparent utility for the hallucinogen in treating addiction. Because we saw a 17.9% decrease within the mice, we may also see a 17.9% similar decrease within humans as well. So my take-home message to you is that first, to teach you what LSD is, it's a hallucinogenic substance that is oftentimes used recreationally, and it is right now being tested for medical purposes in labs as well. Furthermore, I wanted to teach you guys how LSD binds to the 5-HT2A serotonin receptor and why that binding is very important, as well as the lack of knowledge that we have right now based off of the mechanism of how LSD actually works. And it's important that more research is done in order to prove an effective mechanism and application for it as well. And also, it's important in the study of behavioral responses, primarily right now against animals, but eventually in the future, perhaps for humans. Finally, I want to say how LSD could be a promising treatment for alcohol use disorder. As I mentioned before, it showed a 17.9% decrease within mice. So it's important to understand what types of treatments LSD could be used for in the future. These are my references. If you want to check them out, go ahead. And thank you very much.